So yeah, good morning, everyone. Really thanks for attending this seminar talk. Again, my name is Anju. I'm assistant professor who's just joined at Georgia Tech. And today I'm very honored to have this opportunity to present my work at the intersection of machine learning and computational neuroscience. A bit high-level thing is neuroscience studies the brain and its relations to input sensory signals and output behaviors. However, there are some fundamental challenges preventing us from understanding them really thoroughly. So here is a list of challenges people are facing, right? We have billions or even more neurons. We can't record all of them. We don't know many cell types, connectivity, and so forth. But fortunately, advanced neural recording technologies really empower us to dive into and record from our brain at unprecedented scales, and therefore provide us this deluge of neural data and behavioral data for analysis. So although many functions in the brain are still black boxes to us, now we have a large amount of recording neural activity and behaviors, right? And then we can develop some artificial models as surrogate models or normative models to mimic the unknown neural functions. And then, developing machine learning methods could be of great significance to understanding our brain, given that possibility. However, it's really non-trivial to develop these machine learning models. We can't just simply adapt to what we have in the general machine learning field. We need to bring some neuroscience-tailored assumptions to develop more biologically plausible artificial models. And on the other hand, these models can be applied back to the brain data to discover some interpretable results for neuroscience problems. So my work is specifically laid out at this intersection. And more specifically, I'm going to talk about my work from three perspectives. The first one is to discover some latent structure underlying neural recordings. This focuses on how we could understand large-scale neural population recordings in our brain data, but without getting access to some external information such as sensory signals or behaviors. And the second is constructing structural priors for probabilistic decoding for fMRI analysis, where we are also interested in the structures in the brain, but meanwhile, we also want to explore the decoding relations with some external information, such as the visual inputs. And finally, I will switch the gear towards the behavior side and introduce semi-supervised learning for animal behavior analysis and also understanding. So the study of behavior is going to be quite significant to understanding the neural mechanisms and biological functions in the brain. Okay, so let's start from the first one. In a neuroscience experiment, researchers usually have these spike trains like what I'm showing here on the right, and we call it high-dimensional spike train. Since there are a large number of neurons being recorded. By looking at this matrix, it's kind of hard to tell what these neurons are doing. And researchers try to understand some latent dynamics or structures of these neurons. So if we can find some low-dimensional dynamics, we can now explain the behavior of this population of neurons by inspecting this low-dimensional dynamics. There are actually already some existing models discovering latent dynamics or manifolds, so it's not a new modeling topic. But why we still need to build our own model, right? The reasons are, first, we want to build a generative model for population spike train data. And beyond that, we need to incorporate some assumptions inspired from neuroscience or tailored for neural data. And we want to impose some explicit manifold assumption over this latent structure. Moreover, we care about the interpretability of the nonlinear mapping. We model the mapping functions to mimic the functions in real neurons. So next, I'm going to explain our proposed model that covers these points. So here's the model we propose to solve this task. It is called latent manifold tuning model. Here at the bottom is the observation spike train data Y. which is the only observation we have. And our goal is to infer some unobserved low-dimensional process X underlying Y, as well as some probabilistic mapping Py given X. We first assume X is a random process with a prior Px. One assumption we have is latent process evolves smoothly over time, and we can incorporate this assumption into the prior Px. Here we choose to use a Gaussian process prior, which can generate some smooth functions. So therefore, with such a prior, X would be a smooth function of time t, therefore ensuring the temporal continuity. Next, we model this probabilistic mapping Py given X via some nonlinear tuning function or tuning curve f. Tuning curve is usually used to describe the neural functions between some observed input stimuli and output spike rate. But here, the input stimuli are actually unobserved, which are the latent variable x. So when I say tuning curve, I'm basically meaning the nonlinear mapping function to map the latent to the neural spectrums. Given that, one assumption we have is that the output of tuning curves, which are these spike rates, should be non-negative in a spiking model, right? And another assumption is neurons should fare smoothly over this latent space. Therefore, we put another Gaussian process prior over the log of f for each neuron I. This leads to non-negative smooth tuning curves. Okay, so now the output from tuning curves are spike rates lambda, which are continuous non-negative values. Then the spike counts are generated from these spike rates using Poisson distribution for spiking model. So this is the generative model we built to discover some low-dimensional latent process from high-dimensional spectrum alone. And in practice, we reverse the arrow direction and do inference to estimate the latent process X and the unknown tuning curves F given these spectrum data. Okay, so here I'm showing an application of our model to a rat hippocampal dataset. Here's an example of spectrums for 30 neurons in the hippocampus for a 15-minute recording session. These neurons are place cells, which are considered to be able to encode two-dimensional spatial locations of the rat. So again, by looking at the spectrum matrix, it's probably hard to tell there's any pattern of neural activity or what's the underlying structure of these behaviors. And if we do principal component analysis PCA on the spectrum data, we will get at least 10 principal components, capturing a significant percent of the total variance. So this indicates that the spectrum data has a pretty high dimensional linear subspace, which is pretty hard to interpret. For our LMT model, we use the test log likelihood to select the best latent dimensionality, which falls at 3 in this example. This is saying we could find a much lower dimensional nonlinear manifold using our LMT method, which can explain these 30-dimensional neural space. So here's this how this 3-dimensional trajectory looks like. And note that we actually know the rat is traversing in a 2-dimensional W-shaped maze while these place cells are being recorded. And we could plot its real-time 2D trajectory here, right in the middle. We color each arm of this maze for better visualization. And we can see the 3-dimensional latent trajectory actually recapitulates the behavior of the rat in the physical world while segregating the colored arms. Note that we infer this 3-dimensional latent from the spectrum alone without the access to the 2D trajectory. And given the 3-dimensional trajectories, we can find a convex hull covering all the curves to formulate a 3-dimensional manifold. This manifold looks like a folded 2-dimensional W-shaped maze by gathering the yellow and green arms to the center, both close to the sign arm. which is different from their spatial relations especially between the yellow and green arms. We also find the same folding across different recording sessions and different rats. Traditionally in neuroscience, people might usually map the place cells to the two-dimensional spatial location in the physical world. However, we want to ask, can we find some meaningful information in a bit higher dimensional space, for example, the third space? Here I show that place cells actually encode a strong correlation among all three arms with this third dimension. So this is some interesting structure we find underlying these populations of place cells. And one hypothesis is that it might be related to the reward delivery at the end of each arm, which is related to the cognitive function. Okay, so we've shown what the manifold looks like. Next, we want to inspect the tuning curve from the LMT model. Remember, tuning curve is the nonlinear mapping function to map the latent manifold to spike-trained data, right? So we could visualize this mapping function on this manifold for a specific single place cell. We unfold the manifold into a 2D version here just for better visualization, which looks like what's shown on the left here. It shows that this particular place cell fares the most in this circled region. And therefore, we can use this model to well explain the single neuron's behavior with the tuning curve. Here is a quantitative comparison between our latent manifold tuning model and alternatives. We compare the predictive log likelihood on the held out spike trains. The higher the better in this bar plot. We can see that our particular formulation pops out as this best model for this data, giving us a way to explore a whole class of assumptions about latent structure given spectrum data alone. Moreover, I also used the LMT model to find some interesting and interpretable latent structures underlying other cortical areas. Here's an example of visual cortex. I'm not going to dig into details here, just to show that we can actually achieve this rotational trajectory as well as the tuning curve for individual neurons on a three-dimensional torus manifold. And here's another example in the posterior parietal cortex. The mouse executes a perceptual decision-making task with 4 cognitive states. And we discover a three-dimensional butterfly manifold with 4 wings corresponding to these 4 decision states. And we can also obtain the tuning curves for individual neurons on the manifold as well. So lastly, I apply this LMT model to piriform cortex in mice. People don't really have much knowledge of olfactory perception, right? Because olfactory neurons in piriform cortex don't really have this nice spatial topography as visual cortex neurons. And our goal is to discover some latent olfactory topography underlying olfactory perception using calcium imaging data. The data was collected by Bob Dada's lab at Harvard. In order to analyze these calcium imaging responses, I adapted the proposed LMT model previously to a new variant of model to deal with these calcium imaging neural signals instead of spike trains. In the variant, we'll also we'll actually discover some latent topography instead of latent trajectory because we don't really care about the temporal information for this scenario. So here I'm showing the odorant stimuli first before showing the latent and the tuning curve. Our collaborators project thousands of chemical descriptors of these odorants down to a 2-dimensional principal component space. The first PC corresponds to the increase in carbon chain length, and the second PC separates chemical functional groups. And the neural responses were collected by presenting a set of 22 odorants to the mice. This set of 22 odorants actually spans 4 different functional groups, which are indicated by the colors here, esters, aldehydes, ketones, and acids. And within each group, the carbon chain length of the molecule is incrementally varied along the second dimension. And this is actually just a topography of the order set in the chemical space, which is completely inaccessible to the modeling procedure. And all we have are the neural responses given these different orderings. So I first apply PCA to the neural response, and here I'm just showing the 2D plot with the first two principal components. But these PCs are the PC of the neural response, not the chemical space. Here's the LMT estimate. I discovered some interesting latent representations of these orderings, which resemble the true structure in their chemical space. Orders sharing the same functional group exhibit this line-shaped structure in the dataset. Moreover, within each line-shaped structure, the ordering of these orderings is actually also the same as their ordering in the chemical space. Therefore, the x-axis could indicate the functional group now, and the y indicates the carbon chain length. So this provides really nice interpretation for the dimensionality of this latent space. Finally, we can also inspect the tuning curves for individual olfactory neurons to understand how these neurons fare over this latent olfactory topography. Okay, a bit sum up is the contributions of our work here are that we successfully translate neural-inspired assumptions into probabilistic modeling. Moreover, we specifically propose a generative model for population spectrums and also cosine imaging. Second, we impose an explicit manifold assumption over the latent via Gaussian process. Thirdly, the nonlinear tuning curve we propose here is also more biologically plausible with a smooth assumption compared to an arbitrary nonlinear mapping function, say a deep neural network. Specifically, we emphasize on discovering these interpretable tuning curves. It is helpful to understand how individual neurons fare over the latent space, which is lacking in previous works. With the model, we extract low-dimensional manifolds and smooth tuning curves for place cells in hippocampus, visual cortex, and posterior parietal cortex. I also show an application to piriform cortex with a variant of the LMT model. So I am able to find interesting latent manifold and tuning curves for all these areas. And the highlight is really we can discover some interpretable latent structure from high-dimensional neural data alone. Okay, so far I've talked about learning latent variables for multineural data. Now let's switch our focus from multineural recordings to fMRI both signals. There is also some interpretable structure underlying fMRI data that I'm interested in discovering. I'm going to talk about learning structured priors for fMRI data and how it helps for a brain decoding task. A general problem of interest in fMRI analysis is a regression task, which is usually referred to as reverse inference or brain decoding. We have some experimental variable y as the response. And for example, it could be the reaction time during a task or the category of a presented object. We want to map the brain activity x, which is the both signal, to the response y via a decoding weight w. And we are actually interested in estimating this w. We know that usually, although we obtain this whole brain fMRI data, but only small regions of the brain are activated in a specific task. Another observation is that neurons or voxels in the both signal within a neighborhood should be correlated. Therefore, we have the reason to believe that the brain map of the decoding weights should look like something like this, right? With some positive weights, which are the yellow smooth regions, and negative weights, which are these blue smooth regions. Positive weights indicate the positive relations between voxel activity and the task variable, and negative weights will indicate the negative relations likewise. But all the other regions should have no strong activations. Given these observations and the insights about the decoding weights, we want to construct some statistical models to estimate these decoding weight W that can really take advantage of these regularities. So we built a Bayesian model and assume that W has a structured sparse and smooth prior. So structured sparsity means non-zero decoding weights tend to be nearby each other. And this sufficiently reduces a large amount of parameters in W to fit. And smooth means we expect weights within a neighborhood to be correlated with each other. So if we vectorize the brain map of W, so this is just a vector of the three-dimensional decoding weight, this vector should look like this, right? And our goal is to construct a Bayesian prior that can generate W samples like this. So here I'll explain how we build this prior, but first let's only consider the structured sparsity only. The prior encouraging structured sparsity is what we call dependent relevance determination prior, which is DRD for short. We first assume there's a latent variable u, which is unobserved. It is from a Gaussian process prior. As introduced earlier, a Gaussian process is a distribution where you can generate smooth functions from. So such a GP prior can generate a smooth function u as plotted here. And when the mean B of this GP prior is negative, and we exponentiate U to get a non-negative signal G should have some largely zeros, should have largely zeros, but a few non-zero bumps. Therefore, this G function will define the continuous non-zero supports for W. We then formulate a covariance matrix C with G on the diagonal, and this is what it looks like here. Samples from a normal prior with such a covariance matrix C will have this nice structured sparsity. Next, we also construct a smooth version of DRD prior. The covariance matrix now not only has this diagonal bumps, but also some off-diagonal values that formulate the blob for each bump. Then samples W from this covariance matrix will be smooth as well as structured sparse. So this is the generative model we build. Again, in practice, we will infer the decoding weight W and the latent U given the brain activity, X and the experimental variable Y. So now I'm showing an application of the proposed prior to a visual recognition task. The decoding task is to map fMRI data to the category of the presented stimuli Y, which consists of 8 types of objects. So it's also considered as a multi-class classification task. We cast it into multiple binary classifications, and we show that here is the accuracy performance for all binary class pairs at the bottom here. The x-axis is the binary class pairs, and the y-axis indicates the classification accuracies. We can tell that with our priors, we can improve the predictive performance for this brain decoding task compared with some competitive alternatives. So this is the quantitative performance. Then what about the decoding weight? Here's the brain map of the decoding weights for the house versus bottle classification task. Here in the bottom, the smooth DRD weights have significant positive regions in the parahippocampal place area PPA, which is an area known to respond to images of places. And it has some negative regions in the lateral occipital complex LOC, which is an area known to respond to objects. So by comparison, GraphNet, which is a famous regularizer encouraging group sparsity, but its weights in LOC are neither strong nor clustered. So this shows the advantage of our decoding weight using the proposed prior qualitatively. Okay, just to summarize the contributions of this part, I propose a hierarchical model for structured and sparse smooth dependencies with Gaussian precise priors. It improves brain decoding with better predictive performance and also identifies more interpretable brain maps, which is the highlight of this part of the project. Okay, so this is actually another piece of a structured prior work for fMRI data. I just want to mention it briefly given the time limit. We've employed a smoothing kernel in the previous work, right? But it is a Euclidean distance-based kernel structure constructed in three-dimensional brain coordinate space. Such a kernel only cares about stationary spatial correlations between nearby voxels. They assume voxels, if they're spatially close to each other, they should be correlated. But it actually ignores some non-stationary functional correlations, such as the bilateral symmetries across hemispheres, which is the figure in the middle, and also like discontinuities across brain regions. For example, the last figure in the slides, here we have the motor area, which is spatially closer to the mental area, the orange area, but the motor area actually contains some eye movement and orientation information that are more functionally correlated to the visual area in blue here. So later, we propose a brain kernel that encodes such functional correlations. We map the original three-dimensional coordinate space to a higher dimensional embedding space, and then we hope that in this embedding space, the two parietal lobes across hemispheres now could overlap on top of each other because of the bilateral symmetry, and actually they are spatially close to each other now in this embedding space. Also, the green motor area is closer to the blue visual area while being a bit further from the orange mental area. So we think it makes more sense since this motor area includes eye movement and so the proposed brain kernel is also a Euclidean distance-based kernel but constructed in the high-dimensional embedding space. It can better capture the functional connectivity of the brain, and it helps the brain decoding task better with a smoothing kernel, which is really nice. So, but please check out the paper here for more details. Okay. So far, I've talked about discovering structures from neural recording. However, in many neuroscience experiments, neuroactivity is highly correlated with behaviors as well. And therefore, studying behaviors is of great significance to understanding neuromechanisms and biological functions in our brain. So next, I'm going to switch to the behavior side and talk about semi-supervised learning for animal behavior analysis and understanding. So in recent years, there has been an explosion of interest in recording behavioral videos during neuroscience experiments. Often the goal of recording such data is to provide fruitful descriptions for neural behavioral analysis. However, fitting models between high-dimensional behavioral videos and potentially high-dimensional neural data is a challenging statistical problem. Typically, we want to start by extracting some meaningful information from these behaviors by finding an interpretable low-dimensional representation via algorithms such as video tracking, post-tracking, video compression, and behavioral segmentation. Then we can decode some meaningful descriptors of behaviors and have more interpretable predictors for neural encoding or decoding models in the downstream analysis. However, when we have very limited amount of human-labeled data, we can't achieve high-quality behavioral analysis for downstream application. Therefore, in this part of the talk, I will introduce a series of semi-supervised learning models to resolve the scarce data issue by leveraging both labeled and unlabeled data. And this can be able to find interpretable low-dimensional representations from high-dimensional behavioral videos as well. So I will start with the tracking. In a tracking task, we aim at obtaining a bunch of key points capturing the pose of the animal in a video. Animal pose estimation is a critical scientific task in neuroscience. State-of-the-art methods have been proposed to extract the behavioral video traces from these videos. And they've already opened up an exciting array of new applications and new scientific questions to be addressed. A base model that is shared among all these previous methods is as follows. Assume we have a label frame. A label frame here means we have the ground truth body part, locations for this frame, and we input a single frame into a neural network module. This neural network module is the backbone of the post-estimation. And the output of the network is confidence maps for each key point. Here, key points are two-dimensional coordinates of body parts like paw or arm. And the values of the pixels in these confidence maps are from 0 to 1, indicating how likely these pixels belong to the body parts. Moreover, given the two body part locations, we can actually construct some Gaussian bumps for each key point with the peaks at the two locations. Finally, the loss function is constructed to match the output confidence maps to the Gaussian bumps from the ground truth key points. However, there are some limitations of such a base model. The first is it only trains on label frames. However, much more of the information that labels about these images could be found in unlabeled frames as well. So we could include unlabeled frames to enhance the training of the network as well. Second, even with hundreds of thousands of labels, users still often see occasional glitches, implying that the tracking is briefly lost. And these occasional glitches cannot be effectively smoothed out, therefore will typically interfere with downstream analysis of these extracted behaviors. And our solution is to incorporate some temporal and spatial constraints from these behavior videos in order to smooth out these glitches, but during training. So the model we propose is called Deep Graph Pose. It is also a probabilistic graphical model. Let's assume we have a label frame at time t and an unlabeled frame at time t plus 1. We can build the same pipeline for the unlabeled frame here. We just don't know the ground truth for the body part. So here's our solution. We first perform soft argmax with the confidence map to extract single key point location x and y. Then we build a unimodal Gaussian from this 2D location. We use this unimodal Gaussian as the pseudo key point target map to calculate the loss in between the confidence map and the pseudo target map. We call this step as a self-supervised learning step because we don't introduce supervision here. But people might ask, there seems to be no additional information contained in this Gaussian. However, via this self-supervised learning step here, we explicitly encourage the confidence map to be unimodal, which is pretty critical additional information we bring to the model here. And I will show how critical this unimodal assumption is in the experiment later. Now, the supervised learning and unsupervised learning or self-supervised learning parts together formulate this semi-supervised learning framework. Next, we add a spatial constraint to the model. First, let's expand the output to two body parts, which are the pole and arm in this example. For the label frame, we have both the ground truth locations for the two body parts, but for the unlabeled frame, we need to construct the pseudo key point target maps for both body parts using the self-supervised learning step we introduced in the previous slides. Then we can add a spatial constraint in green here to connect the body parts together who actually share a skeletal connecting between for each time point. Finally, we add a temporal constraint in blue to enforce temporal continuity of the same body parts but across different frames. So this is the complete probabilistic graphical model we built. Especially with both temporal and spatial constraints, we could smooth out some glitches that break these constraints during training, functioning as regularizer. Moreover, we hope that the additional links we add to this graph could propagate information from label markers to unlabeled markers. Note that the temporal and spatial constraints are not post hoc smoothing, such as Kalman filtering. We use these constraints to train a stronger post-estimation algorithm. Okay, so here I'm showing the performance on a mouse dataset. We're interested in tracking the 4 fingers on the left paw, and this dataset is a sparsely labeled example with only 55 label frames among 1,000 frames. We compare diplet cut, which is a fully supervised baseline model on the left, with our proposed semi-supervised model, dipgrapos. We see that the outputs of DGP are actually more accurate and robust. To better understand where this difference might come from, we look at the confidence maps for the middle finger from both models. We can notice that the errors in DLC on the left here arise from the multimodality of the confidence map, while DGP outputs a unimodal confidence map, which is due to the self-supervised learning framework. We can see similar comparisons for all 4 fingers here. Here I'm showing the comparison quantitatively. We include an intermediate model DGP semi, which is the semi-supervised learning only without any temporal and spatial constraints. The semi-supervised learning improves the performance substantially, especially in this sparsely labeled regime. And the performance can be further improved even further, right, when we include the spatial and temporal components, as in this full DGP model. Okay, well, we actually also have an extended version of the post-estimation framework for multi-animal post-estimation, but which is under review here. Okay, so I'm done introducing this post-estimation work. Next, we want to combine the tracking information we obtain from DGP tracking with a video compression approach to learn some interpretable latent representations from videos. A baseline model refers to an autoencoder where we input the frames from the videos into an encoder, obtain some low-dimensional latents, and reconstruct the frames using a decoder. It doesn't require the human label supervision as in tracking. It's a purely unsupervised learning model. But the latents of these autoencoder are notoriously difficult to interpret. Individual dimensions of latents may represent many aspects of the video at the same time, but they are also entangled together. Furthermore, the autoencoder doesn't allow experimenters to mark the features of interest if we want to introduce to the model. Therefore, we attempt to combine the strengths of tracking and video compression together in order to increase the interpretability of the low-dimensional representations. So this is the new compression model we built integrating the tracking information from DGP. We introduce a subspace of marker-related latents. So these latents encode the input frames and are decoded to generate the markers, which are the X and Y traces. Now, presumably the other part, the autoencoder latents, should capture variability in the behavior video that is not accounted for by the tracking information. Finally, we can reconstruct the frames from the combined set of latents. Note that this is also a semi-supervised learning framework because we introduced the human label markers into the unsupervised autoencoder model. Given this model, we could ask how well that marker-related subspace is mapped to the reconstructed frames through this frame decoder. The example we take is here the markers are the left pole and the right pole. We first take the latent dimension associated with the X value of the left pole. For example, you can think of these blue dots in this latent space to be the X value of the left pole. Right? And we can move these blue dots up and down, keeping all the other values fixed. We observe that our model has learned a single latent dimension that is mapped to the horizontal position of the left pole, which is the X position. And importantly, there's no vertical movement of the left pole, nor any movement of the right pole. All of that information should be contained in some other dimensions. And we call this phenomenon to be disentanglement. We can actually do similar manipulation of the latents in the marker-related subspace and see, we actually see similar disentanglement across different dimensions. Similarly, we can ask how well the autoencoder subspace is mapped to the reconstructed images. which will also give us some idea of what information that subspace can contain. For the first latent dimension of the autoencoder subspace, you can clearly see the jaw opening and the tongue extending, while other aspects of the image, notably the paw position, stay constant. And the second latent corresponds to local movements of the paw along the base of the holder, which likely captures this small movements of the paw, but which is not accounted for in the tracking information. Okay, so we're able to interpret the autoencoder latents right now. And what we can do with these interpretable latents, we can segment just a subregion of a video, we can decode individual dimensions of behaviors, and we can have more interpretable predictors for neural decoding models. We actually have done all these further analysis regarding behavioral segmentation and neural decoding of interpretable behavioral latents in the paper here, so please refer to the bottom paper for more details. Lastly, I want to briefly mention that we also have been working on some semi-supervised behavioral segmentation work. So again, please check out the paper here for more details. The contributions of these projects are we propose a new probabilistic graphical model for post-tracking, as well as a new video compression method with DGP tracking information. These models are both semi-supervised learning frameworks that leverage both labeled and unlabeled data. Most importantly, they render us these interpretable low-dimensional representations of behaviors, which are beneficial for downstream neurobehavioral studies. So finally, just to remind briefly, this is an outline with all three topics I just talked about in this presentation. As a consequence of this fast development of recording technologies in neuroscience, we are obtaining this more and more complex neural and behavior data nowadays, which will be a norm in the future as well. And we need to break down these two very complicated domains and marry them in a very efficient and impactful way. We could deal with such problems for both system neuroscience with animals and cognitive neuroscience to study humans. And with all these complex data collected, I've shown briefly how to analyze them by building these probabilistic graphical models or Bayesian models, and also the corresponding inferential techniques. Moreover, there are actually all sorts of places where computational approaches can make serious contributions to study our brain. And furthermore, there's also this inspiring direction to develop neural-inspired AI by bringing the knowledge we acquire from neuroscience to build more interpretable artificial models. So there are actually plenty of interdisciplinary research opportunities between neuroscience and artificial intelligence that I'm super excited about, and there's a huge need of researchers to get involved in it. Last but not the least, I want to thank all of my experimental and scientific collaborators. And actually, finally, I want to post an ad for my lab recruitment. So my lab will be focusing on probabilistic modeling and deep learning for neural data, study and analysis. I'm currently looking for highly motivated undergrad and graduate students who are interested in participating one of the projects in the lab. So the first direction is probabilistic modeling for neural latent discovery. I'm particularly interested in developing disentangled and identifiable generative latent models to discover interpretable latent representations from neural populations. The modeling topics would involve various deep generative models such as Bayesian autoencoder, Gaussian process, Bayesian neural network, and etc. The second direction is behavioral analysis and understanding. So we aim at extracting multi-layer information from animal behaviors and allows neural behavioral analysis for both motor functions and cognitive functions. So the research direction will involve projects such as three-dimensional full-body kinematic model estimation, developing hierarchical spatial temporal models for animal behavior syllabus, studying intrinsic motifs and reward representations of animal via inverse or reinforcement learning, etc. And finally, there's another direction about probabilistic modeling and efficient inference approaches that we might try to resolve the scalability and many kind of different inferential problems with respect to Bayesian neural network, deep Gaussian process, deep generative models, Bayesian active learning, and so forth. So yeah, my lab is highly interdisciplinary and demands mathematical skills and experience across multiple engineering and science disciplines. So if you're interested in any one of the above projects and have further questions, please send an inquiry email to my email address. With that, I'd like to stop here for questions, and thank you all for listening.
Applause from everyone joining virtually as well. Thank you, Anchi, for that really, really comprehensive seminar. That was absolutely wonderful. And so right now we're gonna open things up for questions. I know that Dieter Jaeger had a question actually in the beginning part of the talk. Dieter, I don't know if you would like to unmute yourself to ask.
Yes, I would. First, congrats. These are really interesting methods. So I'd hope to have a chat with you sometime soon. It's some stuff that we really, as a mouse behavior neurophysiology lab, really like to maybe explore. My question was about neural activity being smooth and latent variables being smooth. I think that's part of neural activity, but part of it seems to be transient. We have spike transients that come as sensory responses. Sometimes we have transients maybe as error signals. I was wondering whether transients can be incorporated into this model or how they would be reflected.
So the smooth assumption is a really simple assumption we introduced here, just as an illustrative example. When we really fit the model, we don't really impose that assumption only. You can impose different assumption like with dynamic models or manifold assumption, right? It doesn't necessarily have to be smooth. I think if we have prior knowledge about what these, or prior assumption about how these latent dynamic going to evolve with the different dynamic assumption or manifold assumption and so forth, it's totally possible to incorporate them into this kind of generative model. So the smooth assumption is really strong, but it's not really the only one that we can impose. So.
All right, thanks.
Mm-hmm.
I believe Sarah Sola has a question.
Yes, thank you. So this goes back to the first part of your talk, where you show the differences at the very beginning between a PCA analysis and your Gaussian process model. Mm-hmm. So there are two components to this difference conceptually. One of them is that you're comparing a linear method to a nonlinear method. And the second one is that you're comparing a method where you track statistics of the cloud of points versus a method that incorporates the notion of a continuous dynamics. Mm-hmm. I was wondering whether you could disentangle, when you see an improvement in due to the Gaussian processes approach, whether you could disentangle the effect, whether is it due to dynamics or is it due to including nonlinearities in the analysis?
Yeah, so that's a good point. So we do, so the two components are the core things that we want to manipulate for such kind of latent model. And in order to really show the model is working, we compare it to some competitive alternatives, which include the Poisson nonlinear, there is a model called PFLDS. So they have a nonlinear mapping function, but they have a linear dynamical assumption. There is also GPFA, where they assume a smooth prior or some other kind of prior using the Gaussian process over the latent, but they use a linear mapping. So we basically want to see when we manipulate a single component, no matter it's the nonlinear mapping or the dynamic or structural assumption, how much benefit, like both quantitatively and qualitatively, we can get by introducing this nonlinearity over each component. So right. We do see benefits over both. Actually, so the nonlinear mapping function achieves this better improvement way more significantly. So basically, if you don't have a very strong assumption over this latent structure, you just have a nonlinear mapping, that kind of drastically improve the performance a lot by learning more kind of more interpretable latent space already with a better test log likelihood. But as you impose a stronger prior over the latent further step, you're able to further improve that, because that kind of function like a regularity over the latent space that ease the optimization problem a lot. So,
right. Yeah, so I was wondering if you use a method such as, for instance, isomap, whether you would get interpretable latent coordinates, as was shown in the very early paper by Tenenbaum and collaboration.
So yeah, so for the olfactory study, right, we use this double layer nonlinear model to study olfactory perception. We actually compare with isomap, MDS, TSNE, and I think one more popular nonlinear manifold, nonlinear dimensionality reduction method. And only our model can find these separatable like line-shaped structure corresponding to different functional group. The others, they all entangle together. I have that in the paper, but I don't really have the chance to show that in the presentation.
Great, thank you.
Thanks for the question.
Great. Are there any? Oh, there's another question that just popped up in the chat from Shivesh Chowdhury. Would you like to unmute yourself, or should I read your question? Okay, I will go ahead and read it. And so the question is, for the DGP model, what sort of loss functions are used for spatial and temporal constraints? For the disentangle model, how were the latents for markers manipulated to generate mouse moving hand in one direction?
So the loss function for the temporal and spatial are pretty simple. They're just quadratic, because this is the first attempt we have to introduce these two penalties or regularities into the graphic model. But we certainly can go beyond that, right? The square loss means you impose nearby locations to be, for spatial constraint to be correlated with each other, and for the temporal, we hope like the nearby time points should be correlated with each other. This is certainly a pretty naive assumption. The reason we make that is because it's easier for inference purpose. But actually, you can introduce more complicated structures and loss function for that purpose. So for the disentanglement, actually, because we have two components, one is the traditional like autoencoder latent, the other is the marker-related latent, and we map this marker-related latent to the markers. So you can think of it as actually one-to-one mapping. So for example, if we have XY coordinates for a pole, then we will have two dimensions in the marker-related latent space. Each dimension corresponds to, like one dimension corresponds to the X position, the other dimension corresponds to the Y position. So that is how we do the mapping. And then when you manipulate, you just manipulate the latent corresponding to the X position of the pole. And then when you reconstruct the image, you will see if you just manipulate that single dimension, the images will show the pole is moving horizontally. So that's how we do the mapping for the marker-related subspace.
Wonderful, thank you. I'll wait for, and the Shivesh says thank you for your answer. And so I'll see what, maybe another 15 seconds to see if there's any other questions that pop up. All right, well, I think that is, it looks like that's all the questions that are in here. I wanted to thank you again for the wonderful talk, and another round of applause, from the virtual audience. And thank you so much.
Yeah, thank you for inviting me and hosting the seminar.